Hello, welcome to your first lecture of A&P1 lecture. Today we're going to be going over Chapter 1, Major Themes of Anatomy and Physiology. In this two-part micro lecture on Chapter 1, the first one we're just going to look at the history and scientific method. We're going to go over why the history is important, why do I even go over the history, and the main reason why I want to go over it is because I want you to have a nice base in learning of how things came before and how we keep continually changing science, and more importantly, how do we keep changing anatomy and physiology over the years. And then I'm going to quickly look at scientific method. You've all been over it before in high school and middle school, where you look at a hypothesis and then you test for it. But we're going to go over a couple other terms, and I'll explain why those other terms are important when you're looking at journal articles and other things like that. So first, let's look at the introduction. Anatomy and physiology is about human structure and function. We're going to be looking at the biology of the human body. You need to know how the body works. Not necessarily because you're all going to become doctors or nurses, but sometimes it's important for you to understand how your body's working, so if something's going wrong or you're not feeling well, you can help think about and even go to the correct doctor and explain to them what is actually hurting you or what illnesses do you have. So A&P is the foundation for advanced study in healthcare, exercise physiology, pathophysiology, and other healthcare related fields. Most of you are going to be going into the healthcare fields, which is why you're in this class right now. And we're also going to consider the historical developments and a central concept of physiology, homeostasis. So homeostasis and negative and positive feedbacks are two of the key terms we're going to have to memorize in Chapter 1. And if you kind of see, we're going through all of these. Most of this is just background information to kind of give you what's going on in this chapter. You know, study of form. We're going to examine structures of the human body, inspection. We're going to look at. So your lab class, you're looking at a lot of these structures. You're going to be looking at models and then naming them. For the muscles and some of the bones, you'll actually be able to touch them and palpate them on yourself and then feel them. And for muscles, they're really important because when a muscle contracts, it tightens. So you can actually see when you're doing an action of the muscle, it's going to get tight and you're going to know what muscle's working, and that's going to help you during the second part of this course. You can also listen and tapping and listening. Cadaver dissection is important for a lot of pharmacy schools and also for medical schools. Some nursing schools still do cadaver dissections. A lot of them are going virtual, but cadaver dissection is still really important because the cutting, going through the different types of tissue, that's something that you need to learn on a cadaver before you start doing surgery on a human, a living human. And also some of us will go to comparative anatomy if we do the PhD route. You look at other species because other species are easier to test. They still have some of the important structures that a human has, but you can test them on these other animals to kind of see how they would relate in the human. Like a lot of the gene studies that we go through, they're all done in Drosophila or a fruit fly. You've all heard of testing on rats and testing on mice. A lot of people don't have issues with that. So because they have such a well understood genome and genes, you can test on those rodent models and be able to bring them up to human. So this is the first slide of the semester that has something in red, and I want to draw your attention to it. When you see something in red in any of these lectures throughout the entire semester, they're also going to coincide with your study guide. The study guide, you're just going to write down a quick little synopsis of what you see in what's in red. The reason why it's in red is because it's going to either be a direct question on your exam, or it will be part of the question that will help you answer the question. So if you go through all these lectures, even if you're not even listening to them or watching them, if you just go through the PowerPoints and pay close attention to what's in red. Do really good notes with your study guide. Pay close attention to them on the Connect assignments and on your quizzes and discussions. You'll see them over and over again. These are the terms that with this repetition of all these different assignments, you should be able to memorize them and have them down pat by the time you take the exam. But for some of you, you will still need to do some outside work to make sure you memorize these terms with note cards and other stuff. So pretty much in anatomy, we use exploratory surgery. We look and see what the body looks like. Medical imaging is very important because X-rays, sonography, MRIs, these are all things on what we're looking at before we actually have to open the body up. Because when you open the body up, you do have to deal with blood and you do have to deal with infections, you have to deal with a lot of that. So that's why medical imaging is becoming more and more important for diagnosing illnesses and diseases. In gross anatomy, it's a study of structures that can be seen with the naked eye. The reason why gross anatomy is important is because a lot of people don't understand the term at all. But gross anatomy is, can you see it with the naked eye? And this is the key thing that you're going to be doing in your lab class. You'll be seeing the structure of the cell in chapter 3. You're going to be able to see a model in the lab. You'll be able to look at it with the naked eye and then be able to name the structures like mitochondria, Golgi apparatus, and stuff like that. Now, if you're thinking to yourself, I can't see what a cell looks like with my naked eye, that doesn't make any sense. When you start doing the bones and you start doing the muscles, the bones are going to have parts of them that either stick out or go in, and those are all examples of gross anatomical structures because they're very large and you can see them with the naked eye. But for the models, of course, they're just going to be blown up versions of something that's microscopic. But gross anatomy, if you think about it on a bone, if there's something that sticks out, you can see it with your eye, you can touch it with your fingers and your hands. So that's the definition of gross anatomy. Cytology is going to be the structure and function of cells. Histology is chapter 5, and that's going to be what you're going to look at the tissues underneath the microscope. You're going to explain what they are, where they are, and what they do. Ultrastructure, view of molecular detail under electron microscope. Only really large universities have electron microscopes now, and even the large universities are doing away with them because they become so expensive and so difficult to maintain that they usually just send specimens to a company that does it now. And histopathology is microscopic examination through tissues for signs of disease. These are things you would see in a hospital where you would take a tissue. Let's just say you go to a dermatologist, they take a little slice of the skin, they'll look for skin cancer and stuff like that. And with physiologies, there's neurophysiology, subdisciplines, endocrinology, pathophysiologies, and comparative physiologies. But a lot of these are just based off of what are you going to do with your life? And these are all of the different tracks that they have just at Palm Beach State. And if you're looking at these and going through them, nursing, health sciences, pharmaceutical science, athletic training, dental assisting, hygiene, massage therapy, medical imaging, you can get a bachelor's of science now just in medical imaging. And then that you can take directly into a doctor's office. If you're doing just nursing, you can become a registered nurse, an RN, a BSN. You can do LPN, all the way up to an LPN to RN. So there's a lot of different types of things. If you don't even know what those are and what the different levels of nursing are, you can go all the way up to a doctorate of nursing, and they offer that down in University of Miami. So a lot of these things are very specialized for what you want to do. So it's very important to be like, what do I want to do first? Some people just want to do X-ray technician. So they go to X-ray technician, they get into the hospital, and then they realize that I would rather, instead of being the person taking the patient to the X-rays, I want to be the one helping them by the bedside. So then they go back to school for nursing. And because a lot of these are all certificate programs, they don't take a lot of your time. 2 to 3 years at a time might seem like a lot of time when you're 18, but 1 or 2 years at a time isn't that much when you're changing careers to trying to make your life a little bit better. But all of these are all going to be offered at Palm Beach State. So if you're thinking about healthcare field, you can take all of these just at the same college you're already in. So let's get to history. So we go all the way back to Greek, all the way back to Roman. You can go all the way back to the Egyptians if you'd like, because if you think about the Egyptians, they used herbal drugs, salts, physical therapy. They used opium, cannabis, myrrh, fennel, aloe, and thyme. These are all things that you kind of heard about now. Opium for pain, cannabis or CBD also for pain, fennel and thyme, all those herbs that will help with different types of stomach ailments, sunburns, wound care for aloe also. So a lot of these things go way even before the Greeks and the Romans. But it pretty much goes to the Greeks with Hippocrates, or you've all heard of the Hippocratic Oath. Hippocratic Oath is established by this guy and he's called the father of medicine. And he's the first one that didn't really have this idea, but the first one who wrote it down. And he urged physicians to take natural causes of diseases rather than attributing them to acts of gods or demons. So instead of you angering a god, something actually is causing you sick. They didn't know what that was at this point, but he was the first one that says something's actually making you sick. So he's the father of medicine. Then Aristotle came after him, and he's a philosopher that started to write about anatomy and physiology and believed diseases had either supernatural causes or physical causes. And the supernatural ones were theology, and natural was physiology. So he finally separated the two and gave rise to the terms physician and physiology. And he also believed through his writings and speeches that complex structures were built from simpler parts, which was a big deal at this point. They weren't just created out of thin air. And then Galen, he's important because he was a physician to Roman gladiators. And if you know anything about gladiators, you know that they would hack and cut off limbs, have really bad wounds. So he saw firsthand, instead of just looking at textbooks, which they're scrolls and other stuff at the time, he actually looked at animal dissections. And then since the use of cadavers were banned at his time, and because he got to work with gladiators, he got to use what he saw in the animal dissections and see if those surgeries and other techniques that he learned from animals could be transferred into humans. And he saw science as a method of discovery, not just a body of facts taken on faith. And he wrote many books. And then he started to advise followers to start looking at observations, then looking at religion and other things that would cloud their judgment. Then there's also Christian culture in the Middle Ages. They just suppressed science. So a lot of the progress at this point was in Jewish and Muslim countries. There was a Jewish physician, Mennonites, and he wrote 10 influential texts. He was a physician to an Egyptian sultan, Saladin, and all of his texts were also used in the Muslim world by Ibn Sina, and he's the Galen of Islam because he started combining Galen and Aristotle and other works, and he wrote a lot of books about medicine that's still used in medical school for about 500 years until the Renaissance. And then we're now in the Renaissance. We're rethinking everything, and Vesalius was very influential at this time because we start using the same type of techniques he used to this day. So during the Renaissance, the Catholic Church relaxed all their restrictions, a lot of their restrictions, not all of them. Barbering and surgery were considered kindred arts because they both used the knife. You probably wondered why a barber pole is striped, because what they did, they'd have a pole on the outside, and because they used knives, they would cut off abscesses and other minor surgeries in barber shops back then. They would have a lot of towels that were bloody, and when the wind blew, they wrapped them around the pole. So soon the barber pole, that red and white spiral, became their signal. And Vesalius published the first atlas of anatomy. And that might not seem like a lot, but after he did it, Michelangelo and other artists and other humanists started to write down and pay really close attention to how does the body look. And because he started looking at things, they started looking at different muscles, they started looking at bones, they started looking at what's pumping blood, where is knowledge coming from. And they started doing this by looking at Atlas of Anatomy. And you're still using Atlas, you're still using pictures in your lab textbook. You're going to be using techniques that he discovered back in 1543. And then William Harvey and Severus, this is much later in the 1600s. They started looking at different things. I mentioned that the heart, he realized blood flows out of the heart and back again with Harvey. Severus also started looking that blood must circulate continuously around the body. At this time, they thought it went into the tissues where it ended, and then it was reformed again someplace else in the body. So even in the 1600s, they still didn't understand blood circulation. They didn't really understand cells at this point until Hooke had a microscope and actually looked at how cells are made. And this is a very famous picture of just cork. It's a bark from the cork tree. And he was able to see that these little things look like little cells in a prison. And that's why, to this day, the building blocks of life are called cells, because of Hooke and his microscope. And then these two guys just picked up, they made better microscopes. They were looking at different things, and they were actually able to find more structures and trying to see things. Carl Zeiss you still see to this day because of his, in Germany they still make lenses named after him because of how important he was with optics. And then Schleiden and Schwann, you'll hear a couple things named after these two gentlemen because they were scientists and doctors at the time that started to name things. Because they started seeing cells, they started to name them after themselves. And up to this point, they concluded all organisms were composed of cells. And those cells work together to do something else. So in modern biomedical science, technology enhancements, if you're in your 30s, you probably have been alive long enough to realize how much MRIs have changed. In the 90s, you couldn't get an MRI. They were too new, and they were something that you would only get if you had really good insurance. Now you can walk into a place on the corner that has a diagnostic center, and they can get an MRI. Most hospitals offer them, and they're very quick and they're very easy to get. And the imaging that they're able to do now is much more efficient than it was even 5 years ago. Also, the genetic revolution also in the 90s. So in 1999, they were able to get the entire human genome. It's called the Human Genome Project, if you want to look it up. And they're able to see what were all the bases that made up the total human genome. And now after that, they're able to look at the genes and how they're set up and see them to treat different types of diseases. We'll talk about that when we talk about DNA and RNA. And that's in chapter 4. And then all of these early pioneers were important because they established a way of thinking. And they replaced superstition and religion with actual natural laws. And you're probably thinking, is that important? Yes, because even around the 1800s with Francis Bacon in England and René Descartes in France, you're going to start seeing a lot of French and English scientists now because of those two governments, they started to give money to scientists to start researching stuff on the human condition. And even today, a lot of the scientific research that happens in the United States happens through the NIH, or the National Institutes of Health, and they are actually driving where science is going in this country. So that was set up about 200 years ago. And then Francis Bacon, numerous observations until one becomes confident in drawing generalizations and predicting from them. If that sounds like something you've heard about, you would look at something, you do research on it. You might even watch a bird fly around, and then you can predict something from it. And that's the beginning of making a hypothesis. And then if you make a hypothesis, then you can test it. So Francis Bacon started thinking, how in science are we supposed to test things? So the Pato-deductive method, more physiological knowledge gained by this method than any other method ever designed, because the investigator or the person or the researcher asks a question. I think this will happen. So he formulates a hypothesis, and then he's going to test it. And the better you get, the more knowledge you get, you can start making good hypotheses. It can be consistent with what is already known, and then it's testable and possibly falsified with evidence, falsifiable with evidence. So if you kind of know what's already done, you build upon it, and then you test it and see if you have actual evidence to either make it true or false. And you do that by different things. Sample size. Sample size is important because if you have too little, you don't have enough to actually make a good judgment. That's why a lot of political polling is bad, because they might use a sample size of 1,000 or 10,000 or 20,000, doesn't matter, but they'll say an entire state's going to vote one particular way. And even if you have a sample size of 1,000 or 10,000, most states are a couple million. So it's very, very difficult to get good research if your sample size is too small. Also, how do you do the controls? If you're trying to test if a drug works, you have too many different things that can be tested. So setting up a good control group is very, very, very important. And then psychosomatic effects. You probably have heard about this with antidepressant medication, that a lot of these types of drugs, they have something called the placebo effect, where if you think you're taking a drug, you'll start feeling more healthier. And a lot of the things with the brain becomes a problem with the placebo effect, because people start to believe they're feeling better, and those are only short-term benefits. And then, almost done, experimental design. The experimental bias. Sometimes you'll want to do a double-blind study where the researcher will write down, this one is the drug, A is the drug, B is not the drug. Then they will give that to another person. That person will then schedule a group of people, usually 3, 4, 5, depending on what type of project it is. That person who's scheduling the research doesn't know if A or B is the drug. So that means the people who are testing it, either rats, mice, or cells, they don't know which one is going to be the drug, which one is going to be the control, or saline water if it's a drug. So they can't bias the study. You can't lead your research in one way or another. And then also statistical testing. There's a lot of different types of statistical models that you can use that help depend on fit and see if what you tested is scientifically reliable. And then one thing, peer review. This gets a lot of people into trouble, because you listen to a podcast, you listen to somebody's opinion on the news. It's just that. It's somebody's opinion. It's not a fact. It's not something that they've researched. It's something that they kind of saw or they read, and then they are now trying to tell you that it's fact, but because it's just their opinion, it will always remain an opinion. If you're reading a journal, most good scientific journals are going to use peer review. And these are going to be experts in the field that start to give feedback to that scientist on how to make their project their research project or their testing better. If you're not an expert in the field, they're not going to ask for your opinion. And you might say that's kind of insulting, but this is good, because unless you're an expert in memory and learning, you shouldn't be giving your opinion, again opinion, to a scientist that is an expert in that field. Other experts in that field will see their research methods, they'll see their numbers, they'll see their the way they were running a study, and they will come up with unique insights into that testing that a non-expert wouldn't do. And another thing, if you are doing independent research and you are looking for stuff, especially if you're going to do a master's or a PhD, you're going to start looking at how journals are ranked. And a lot of journals are ranked by impact factor. An impact factor is very, very, very important because it will tell you how important or how many people read that particular journal. And you might think that that's not that important, but the higher the impact factor, the more likely the stuff you are getting from that journal is correct. It's been fact-checked, it has been peer-reviewed, everything in it is at the highest quality. There's a lot of journals now that are just online journals, and they can publish pretty much whatever they want. And just like your news at night, if you watch the news, it's very difficult to determine if it's somebody's opinion or if they're giving you fact. Those online journals are there for more entertainment, just like a lot of those late night shows are. They're for entertainment, they're not necessarily for facts. And then finally, scientific fact is something that can be independently verified. You have laws of nature. These results from inducted reasoning and repeated observations. So a law of nature would be like the law of gravity. A theory, on the other hand, is an explanatory statement or a set of statements derived from facts, laws, or confirmed hypothesis. So we don't know if they're actually true because it's a theory, but we're going to keep studying it until we can make it into a law or a fact. And this is the first part of 2 micro lectures for chapter 1. Keep going, and if you kind of remember, there was only 2 things in red. Vesalius, the father of medicine that made that first atlas of anatomy, and the other one was gross anatomy. So if you kind of remember those 2 terms from this lecture, everything else we went over is just a building block to move you a little bit further throughout this class.